and welcome to the Research Matters podcast. I am your host Aarti Halve, and today we have with us Dr. Chandrasekhar Nair, awardee of the Infosys Prize for Engineering and Computer Science 2021, and Professor Arvind, chair of the jury for the Infosys Prize for Engineering and Computer Science 2021. Dr. Nair is the co-founder and chief technology officer of Molbio Diagnostics, one of India's leading healthcare companies in the in vitro diagnostic segment, and the manufacturer of the renowned molecular diagnostic platform TrueNat Real-Time PCR. He is also the founder of Big Tech Labs, a fully owned subsidiary of Molbio Diagnostics. Professor Arvind is Johnson Professor of Computer Science and Engineering and the head of Computer Science faculty at Schwarzman College of Computing, Massachusetts Institute of Technology. He is a renowned computer scientist whose work has been instrumental in the development of dynamic data flow architectures and associated parallel programming languages. Dr. Chandrasekhar Nair has been awarded the Infosys Prize for his development and large-scale commercialization of TrueNat, a new point-of-care testing platform for PCR-based medical diagnostics. Dr. Nair's work has enabled testing for millions of COVID-19 cases across resource-limited settings in India and the diagnosis of many infectious diseases, including tuberculosis, worldwide. Hello, Dr. Nair. Hello, Professor Arvind.
Thank you.
First of all, congratulations, Dr. Nair, on the Infosys Prize.
Thanks, Aarti, for your good wishes.
My first question is to Dr. Nair. What was your motivation for developing the TrueNat real-time PCR platform?
To me, very straightforward thing that we wanted very good diagnosis, especially coming from India, living in India, having gone through so many infections as part of your life. You want to not be in a situation where there is no definitive method of identifying what is wrong with you. Because once you can precisely identify what is wrong with someone, what is ailing that person, you can then have a very directed therapy and a very rapid return to wellness. And as far as infections are concerned, this is also very important to prevent the spread of infections.
Correct.
So this is what led us to start working on the PCR. Lead us to start working on the PCR. PCR, as you know, is a technique that has very high sensitivity because you are amplifying the signal many times because of amplifying the DNA itself and or the targeted DNA segment itself. And it also has very high specificity because by technique, you will only amplify pathogen DNA. So therefore, it is possibly among the best tests that can be used for pathogen detection. So that is the reason why we decided to work on PCR. Now, a country like India, where we considered as one of the typical use cases, does not have the infrastructure required to run a PCR. And when we conceived this in the year 2002, 2003, that was a very different era where I think in a city like Bangalore, we possibly had one PCR machine in the clinical setting. And if you wanted to take PCR to point of care, like what we wanted to do, it required to re-engineer everything, but that's a different part of the entire story. But the motivation was that to provide precise diagnostics at point of care or where needed the most.
You mentioned that we did not have the kind of infrastructure required for the PCR. What was so complex about it, and why or what all is required, maybe?
So the first thing is that you are handling a potentially infectious sample. So there is a biohazard in handling the sample itself.
Right.
Secondly, the sample using any biological matrix, whether it is blood or whether it is a swab, as in the case of COVID, or whether it is sputum, as in the case of tuberculosis, all of them are inhibitory to PCR. And therefore, you require to separate out everything other than the nucleic acids of interest, which you're going to amplify. So the preparation of the sample required, let us say, very careful handling, biosafe handling, and also required expensive technology. The third part was the PCR itself. And these PCR machines were huge, bulky machines that cost a lot of money. I remember we purchased our first PCR machine for 35 lakh Indian rupees. So that is not something that
Oh,
could be used for running PCR in the clinical setting. And
right.
all the reagents were stored at -80 for long-term
Wow.
stability. So, and you really require to have a PhD to run a PCR because there were so many things that could go wrong.
Oh my
So the
God.
idea was to try and eliminate all of these errors and make something that would be applicable for point of care settings, addressing each
Mhm.
one of these requirements of training, of infrastructure, of cost, because the PCR machine typically runs 96 samples, and
Mhm.
that means you will have to club all of these 96 samples to even get something reasonable in terms of cost, because the
Mhm.
reagents required were very expensive and were very fragile, very heat labile, etc. And
Mhm.
all of them resulted in very high cost testing. So we had to rethink the entire uh,
Uh,
thing. And when I look back at this, I think we were a bunch of crazy guys to attempt something like this at that point in time. But okay, I think
Yeah,
it was good that we did not know much.
yeah. I think one needs to dare, that is one important thing that I see in this enterprise. Uh, So what you have mentioned some challenges that you had in front of you, right? Like the cost and the complexity. What were the specific engineering challenges, and how did you overcome those, or how did you manage to work through, around those or work through those?
So we had a problem that we wanted to run PCR at point of care. And then
Mm-hmm.
we try to create a solution. That solution typically required a multi-pronged approach. So there was no engineering, no biology, no chemistry, no computer science, everything into one. We had to
Okay,
solve this problem, and
correct.
we solved it using very unique methods. One of the methods that we solved the sample preparation with was using nanofibers. We initially started with
Okay,
magnetic nanoparticles, and we used to import those nanoparticles. And the cost of one test with the imported nanoparticles was, I think, three times the cost at which I'm selling the PCR test
oh
today.
my God.
So that was not something that was feasible, and it was also
Mm-hmm.
not something that was possible to automate very easily because it required moving parts. We wanted a point of care solution, so we wanted a rugged platform that would not fail
Mm-hmm.
very easily in the field. So we moved to nanofibers. We immobilized these nanofibers in a plastic cartridge, started with
Mm-hmm.
500 microliters of the sample and ended up with 100 microliters of pure DNA. Now, this DNA was pure
Okay.
enough to do, and we know now today that it is pure
Mm-hmm.
enough to do even sequencing from the DNA. So,
Okay.
extremely high quality DNA, but the requirement was that of a microfluidic cartridge. Now, this was not something that was ever available in scale. And when people talk about microfluidics, they typically talk about 1 or 2 microliters. But
Okay.
we did not, and this is a question that I got asked a lot when Theranos was very popular, saying that they are
Uh-huh.
doing everything out of a few microliters, and you want half an ml of sample, etc. But
Mhm.
I was not able to answer that question very effectively at that point in time. That okay, there was only so much pathogen that you could have in 500
Mhm.
microliters of a sample,
Mhm.
and if you have only 5 microliters of the sample, then your chance of finding that pathogen in that 5 microliters is going to be significantly lower. So hence your
Right.
sensitivity cannot be what Theranos wanted to do. So ultimately
Mhm.
with biological samples, you have to start off with a larger volume, but reduce it to a lower volume. Now, how do you do this in practice? It required us to have precision plastic engineering and then the ability to
Yeah.
bond these two pieces of very flat plastic together and do this in very large quantities and do this at extremely low cost. That was a
Okay.
massive engineering challenge. And
Right. Right.
similarly, the PCR itself was typically run off a PCR block, and the
Okay.
block was a highly machined alloy, that allowed you to heat very quickly and then dissipate the heat quickly to a heat sink.
Mhm.
But you used to heat all these 96 samples together. Because we wanted
This.
to do this at point of care and we did not want to batch samples, we wanted the samples to be run as and when they came in. We decided
Mhm.
to run it off a single well. Now the question was, what is the construction of that well?
Mhm.
And we started with silicon. And then we broke our heads against a lot of the silicon foundries that existed in India, the facilities that existed in India, and we gave up. And we said that this is not
Okay.
going to happen unless I go to, let us say, a high-tech silicon foundry outside the country. And while silicon is great to work with, and once it is in very large numbers, it can be at really low cost. The cost of development was not something that we could afford. So we decided to look at other materials, and hence we went back to the basics. We said, what is the best material to work with DNA? And that was glass. So, and then we started
Oh.
looking at glassy substrates. And hence we came up with ceramic. And then we came to low temperature co-fired ceramic. And then engineering that low temperature co-fired ceramic in a fashion that gave very uniform temperatures, because temperature artifacts are one of the biggest reasons why PCR results could go wrong. So you required absolutely identical temperature in that entire volume of sample. We reduced the volume to 6 microliters so that you could do this very rapidly. And theoretically, I can do a PCR in less than 10 minutes today with the ramp rates that I can go to with this MEMS inspired chip. But
Mhm.
we don't do that because we know that the biology typically lags and we are not in a race. Ultimately, we have to get the right result out.
Right.
So that was an engineering challenge itself. Scaling that up in India was an
Uh,
interesting challenge. There were so many
So.
things. I think when we look at it, the chemistry, stabilizing the chemistry such that it would be room temperature stable,
Mhm.
then building the entire support infrastructure around this, because we are talking about point of care. So how many point of care
Right.
solutions are out there, complex point of care solutions? And when you're
Right.
deploying in the thousands, how do you ensure that your platform, in the COVID second wave, our platforms were running 24 bar 7.
Right.
And that was kind of an absolute thrill for us to see that something that we developed had the ruggedness to be running. Technicians were running in 3 shifts and they were running these platforms 24 bar 7. So the entire support infrastructure required, the IT required behind this to build enough intelligence to ensure that you don't require an expert at the end of the line to look at the results.
Mhm.
The IT required to run that entire platform, the intelligence required to run that entire platform, encapsulated within that platform. All of those things were challenges in their own right. But
Right.
we just looked at this as a multi-pronged problem and went ahead and started looking at solving them one by one.
Yeah, that's some really huge work. How long have you been working? I mean, from when you conceptualized till you got the product out, what was the time span like?
So we started as Big Tech Labs in the year 2000, and we said
Mhm.
that we want to do diagnostics at point of care. We did not say we want to do PCR at point of care, and we thought
Mhm.
that we will do blood glucose measurement at point of care. And just
Okay.
to put it in context, we were a bunch of scientists with a scientific background. We got a lot of other people, like-minded people with us, and we started looking at glucose sensing, and we came up with a design that could do very accurate glucose sensing. Then
Mhm.
we found multiple problems with that approach. First of all, the blood glucose in the body does not require that degree of accuracy unless you're going to act on that information. If you have an
Right.
insulin pump, then you can say at this particular level, I will start pumping insulin. But otherwise, the body's blood glucose varies so dramatically that you don't
Right.
require something that can measure the glucose to plus minus 1%. We also
Right.
realized that while there were no products of the category that we were talking about in the market, that space was very crowded as far as IP was concerned, because there was a lot of IP already. So freedom
Right.
to operate would have been very difficult. So we decided to move away from doing biochemistry to doing
Mhm.
PCR. And that was a very quick and very easy decision, because I think all of us said that, yes, we now know what to do. So what is the
Right.
problem that we should tackle? The problem was that of infectious diseases. So we pivoted to the PCR and started working on the PCR in the year 2003.
Okay.
2005 is when we had a lab level, very, very, very early concept that we could show to the CSIR NIMITLI program, the New Millennium India Technology Leadership Initiative program.
Mm-hmm.
And they not only funded us, they gave us a soft loan, but they also got some of the best minds in the country to kind of guide us. And that was, I
Mm-hmm.
think, the turning point for us. So you can easily say that 2005 was that formative year for us to start working on the PCR in full force.
So you mentioned two manufacturing problems here, right? One was the plastic that you said, right, where you for the cartridges. Is that correct? Did I
Yes,
understand that right?
yes.
And the glass,
The ceramic
right?
substrate for doing the PCR.
That was manufactured in-house, or
So we made
you
the
had
first
some?
versions in-house in our own lab, and that
Mm-hmm.
is what gave us the confidence that we could do this in precision manufacturing, large scale. Of course,
Mm-hmm.
now it is done in very large facilities, but this is possible
Okay.
to do in the lab, and we did our first devices in our lab.
And then for the production, it is outsourced, or
Currently outsourced, but we are trying to bring that also into our own
okay.
manufacturing facilities.
Okay. So let me put the next question to Professor Arvind. After hearing this whole journey, I it makes I'm curious about on what salient points did the jury select Dr. Nair for the prize?
So we were quite aware of this journey. And I think sometimes people in India do not appreciate how much engineering is needed to bring something to a usable level or to market and so on.
Mm-hmm.
So this was like a perfect example for us, that there is
Mm-hmm.
science, you can do it, but that's a very different story than actually doing it at scale and bringing it to market. And there were, as Dr. Chandrasekhar described himself, so many challenges involved,
Right.
and he tackled them one by one. So that was very, very impressive for us.
Right.
And we really wanted to recognize such activity because I think there is much more activity of this sort needed. People think scientists are needed only for fundamental research. Scientists
Mm-hmm.
are needed, scientists and engineers are needed at every level of development. And at the same
Right.
time, it was very useful. And we were also aware of the fact that it was almost like a race because he has friends in West who were trying to do the same thing, right? And
Hmm.
he's the one who succeeded, right?
Right. Right.
So
Uh,
it's really truly an international level accomplishment, which has special
right.
bearing for India.
Yeah, science, it takes a few steps to go from a scientific concept to, a marketable product and actually sell it. So,
Exactly,
yeah,
exactly, right? I mean, it's one thing to produce one automobile. It's completely a different story when you want to produce, 10 million automobiles a year
right, right,
with very low defects. I mean, that's a
right.
completely different level of engineering and requires lots of fundamental work. Actually, I was curious, Dr. Chandrasekhar, for the development of cartridges, these plastic cartridges, did you bring in outside expertise? Did you collaborate with somebody, or you ended up doing all of it in-house?
We did all of this in-house because we actually went to multiple people outside and they would work with it for a period of time and then they would give up.
I see, I
For them, it was very easy to give up. They just had to say, no, it can't be done. I
see.
had no choice. I had to make it happen. So we first
This,
started with ultrasonic welding and they said it could not be welded using ultrasonic welding. Then we figured out that if you use a different frequency, it will weld. Then we found out that ultrasonic welding also gave rise to microchannels around it. Microchannels came with their own complexity, so we had to figure out a way to solve that. Now we moved to laser welding, and I think, we're one of Asia's largest laser welding facilities at Vizag today.
and
Oh,
I was thinking that this cartridge may be another side business you can start.
I think we
Because
understand plastic
many other
bags now. Well, yeah.
So each small element in that whole thing was some discovery, some achievement there.
Some things that you take for granted, possibly in the West, were
Right.
huge challenges for us. To get a plastic piece that is extremely flat, that I can
Mm-hmm.
bond together, was such a major challenge. And I burnt my fingers with so many different people who made the molds for that, that it is not funny.
Oh.
And uh, I mean, there were times when you just wonder what is going to happen because everybody promises to do something, and then 6, 7, 8 months down the line, after a lot of follow up, you hear that this is not going to happen.
No,
So
and
we did
maybe
something.
you'll.
I told you this is one of the things that I didn't do smartly. This is something that I should have done much more smart.
Uh, but maybe they genuinely can't do it. That is even more frustrating. I mean, maybe they are trying, but they are not able to
Yes,
do it.
they
So
could
that is
genuinely not do it.
right.
And the reason for that was a lack of the ability to do precision engineering. I think we are very
Mm-hmm.
happy doing non-precision parts, but when you have to have a very high reproducibility across millions of parts, we fail miserably as part of our manufacturing ecosystem. Our ecosystem was
Okay.
very primitive at that time. I think our ecosystem is also catching up now, and I should say that I'm super happy that there is an effort being put to improve that ecosystem, without which products will not come to market very easily.
So, you mentioned that you started with tuberculosis, right? So, how easy or difficult was it to extend TrueNAT to other diseases?
So we actually started with hepatitis, and I should tell you I love the blood sample because it's very easy to purify. And then we started looking at the sputum sample, and I'll tell you it is the most difficult sample to handle because sputum sample varies so dramatically from person to person in terms of its physics, in terms of its composition, in terms of what the person has eaten, if the person is a smoker. All
Right.
of those things affect the consistency of the sputum sample, and it varies so dramatically with geographical conditions. So in the winter, it is much more thicker. In the summer, you have a thinner sputum, and you are supposed to take a sample like this, homogenize that in the field, causing absolutely no aerosol, so that the person who is homogenizing is not affected. Take 500 microliters of that and process it to get DNA. Once you do sputum, I think you can handle any other sample, even stool samples for a breeze.
Oh my
And there's
God.
some people
Okay.
eat pan also, right? So
Yeah. Oh, yes, exactly. You have chewing tobacco, you have pan, you have blood in the sputum, which are produced by people with TB. And
Oh.
other solutions,
Oh.
including Gene Expert, that is the other WHO approved molecular test for TB, fails if there is blood in the sputum sample. But by that time, we had significant expertise handling blood. So we were able to handle that. So the beauty of the prep is that our prep does not change. The cartridge does not change. The reagents don't change across
Oh.
all sample types. The only thing that changes is the buffer that you collect your sample in. And I think that is a very massive chemistry breakthrough. I don't think anybody has been able to do that.
My next question is to both of you, Dr. Nair and Professor Arvind. Uh, how does having an accessible testing facility impact the handling of a disease from a public health perspective? Like, what advantage specifically does having a point of care testing facility have on controlling the disease or giving the patients treatment or things of that sort?
Professor Arvind, you want to go first?
you go first.
Okay, I'll just answer that with a very simple example. We had an order for 1,512 machines for the National Tuberculosis Program during the COVID
Mhm.
first wave. In fact, the order came in February. In March, we had
Okay.
our test validated. In April, there was a pressure for us to deliver all of those 1,500 that were supposed to be delivered over 1 year immediately because COVID came into this country.
Oh,
And that when we had our test released, we moved from having only 20 testing centers in the country that was ICMR approved to
Right.
having 1,512 plus 20 centers in the country at the click of a button. So that is, and all of these were at point of care. These was at smear microscopy centers across the country. So I think the impact that we had in terms of scaling COVID testing was unbelievable. We
Right.
did nearly 12 million COVID tests last year on the platform, and that was 12 million tests that could not have been done if you did not have access to point of care testing.
Right.
The greatest advantage for me and the greatest joy for me is that 5 kilometers from my village in Kerala, you can do COVID testing on the TrueNat platform.
Wow. Right.
No, I think it
Yeah.
makes a huge difference in everything. And this is one of those things that it's hard to imagine how it affects you until you see it. So, for example, I'm in an environment where uh, MIT provides me facility for, actually, I'm required to test once a week, and all I have to do is just give my swab, and the results come back 12 hours later. But it'll make a big difference, if the result came back, half an hour later. So point of testing really does make a very big difference, you know, in many things in administration and so on. I think if you're COVID positive, then you would like to get on with it as soon as possible, right? In terms
Right.
of isolation and sequestration and so on.
Right. Right. When I was reading up, I found that there's a mention of reducing the tuberculosis detection window to 3 weeks instead of months.
Absolutely.
So I mean, is it possible to explain what that means? I mean, I was
Yes,
not able to.
absolutely. See, tuberculosis, the gold standard for diagnosis was smear microscopy. Now, the problem with smear microscopy is that your sensitivity is only about 50%, which
Okay.
means that you will miss 50% of the people who come in for smear microscopy. You will misdiagnose them as being negative.
Okay.
The reason for that is it takes 10,000 acid-fast bacilli per ml to be able to detect tuberculosis using microscopy.
Oh.
Clinically, it takes 6 months for you to have 10,000 acid-fast bacilli per ml. You start coughing maybe in the
Oh.
first week of active infection with TB.
Mm-hmm.
So when we cough, we are not going to keep quiet, isn't it? We're going to go
Right. Right.
and get a medical opinion, and usually we'll get an antibiotic thinking that we have a bacterial infection, which we do.
Mm-hmm.
That antibiotic would not be useful. So 2 weeks later, you go to the doctor will prescribe some other medication for you.
Right.
And this goes on, and then sometime down the line, you are becoming sicker. You are starting developing night chills, fever, and
Mm-hmm.
the clinician may say, oh, maybe if he's a good clinician, and say maybe we should test for tuberculosis. And the
Mm-hmm.
test that is available is only sputum microscopy. So the microscopy test will say you're negative.
Oh.
So the clinician will continue giving you different and more powerful antibiotics. And that is not enough because it is not directed therapy. It is a very short therapy. And in
Mm-hmm.
India, somebody like me who has access to good medical care have even had fluoroquinolones for a cough.
Oh.
Now, fluoroquinolone is our last line of fight with multi-drug resistant TB.
Oh.
Okay, but you have to take it for a period of time. My
Mm-hmm.
doctor will prescribe 5 days of fluoroquinolone for me and I'll be fine. And that is the same case with the TB patient. But
Oh.
that patient will come back after a few weeks with significantly worse symptoms. And he may be
Oh.
drug resistant by that point in time. This is the reason why TB has never been eliminated. TB is a
Right.
part of our civilization. I
Oh
think you
my
have
God.
it in Egyptian mummies,
Yeah.
you have it described in the Rig Veda. So this is a bacteria that has kind of co-evolved with humanity. And the only way
Mm-hmm.
to eliminate this is by diagnosing it early.
Okay.
The PCR test has a limit of detection of less than 100 cells per mil. And that load you will
Okay.
get in the first 3 weeks of cough.
I see.
Okay.
I see.
So
Okay.
you will be able to diagnose tuberculosis positive if that person has had symptoms for 2 or 3 weeks using
Mhm.
Mm-hmm.
a very sensitive test, which is what the PCR is. So that is
Oh,
how we are reducing the window from multiple months. In fact, there are reports that it takes an average 3 years for a person to be diagnosed with TB. Those
my
3 years,
God.
not only is he suffering, he is also
Right.
spreading the disease. So that is
Right.
the issue.
So that's a
Yeah. So
very clear example of early diagnosis making a big, big difference.
right, right. So that sort of leads to the next question that I have, that is, how can use of technology impact the diagnosis or the vaccination against a disease or the treatment of a disease? I mean, all of these three. So this is again for both of you, your thoughts on this, the impact of technology on disease management.
I think these are slightly different things. I mean, like this COVID vaccine development has had such huge impact that now
Mm-hmm.
people are getting to a point where they are ready for next uh, very large number of viruses, because they see similarity and I think next time the vaccine development will be even faster, in this
Mm-hmm.
case. But that is still, even if you have vaccine, you still need early detection, right? And you need
Right.
cheap detection because if the detection is very expensive, then itself it becomes an impediment,
Mm-hmm.
for that.
The future of managing diseases and pandemics, I think we will have much faster vaccines, much more effective vaccines developed. We'll have
Mm-hmm.
much more faster diagnosis. So we will be able to diagnose and take appropriate measures to prevent the spread of disease. So I think
Mm-hmm.
all of these will lead to prevention of these large scale disruption of life because of pandemics. And that is what technology will actually eventually help us, is what I would believe.
Right. I would like to have opinions from both of you. There is an increasing use of artificial intelligence and machine learning in all fields. So, how do you think AI and ML can help in the medical field? Or, I mean, I guess it must already been used in the medical field. I would like to understand in what way it helps and it would help.
If I may take a crack at it, I
Yeah.
think one way to think about AI and machine learning is how to make use of data for
Mhm.
drawing better conclusions. So
Right.
until now, the way science has been done is primarily we have theories and models and we try to explain things, but you can invert that process. You can say, my gosh, I have looked at 10,000 TB patients, right? I mean, how
Mhm.
you classify them is a slightly different matter, but you can say, I've seen 10,000 people with similar symptoms or something. And
Mhm.
then you can feed that data into machine and apply machine learning techniques to detect commonality, to detect predictive power of it. So
Mhm.
you can say, oh, now if you give me a sample, tell me something about something, then I may accurately be able to tell you whether that person has TB or not. I
Okay.
didn't say
So,
it very well, but maybe you got the
right.
right. So first
Right.
condition in this is that the sample has to be in some sense representative of the population.
Right.
And you can draw a distinction. You can do machine learning in a blind fashion where you know nothing about the problem. You're just trying
Mhm.
to deduce something from data, right? But there is much more interest in doing machine learning when we already have some knowledge. So we may know a lot about tuberculosis. So
Mhm.
we like to use machine learning algorithms which are cognizant of those constraints or those features that we already know about.
Right.
And once you start doing that, then the accuracy improves and people feel better, about it
Right.
because sometimes machine learning goes wrong in very strange ways, which
Right.
throws off people. But so I think machine learning is going to be used in everything. I mean, let me explain it from a different point of view. When I was in IIT in 60s as an undergraduate, we
Mhm.
learned very fancy theories, for example, how the ocean may be turning, right? Is
Mm-hmm.
ocean circulation models. But we have zero data, so we do not know if this theory is right or not. So if you move the needle, 50 or 60 years later, those theories haven't changed one bit, so they're probably right. But now we
Mm-hmm.
also have tons of data, right, which is being collected from satellites and this and that. So now you can really see if the theory works, right? Or if it
Right.
doesn't work, how it should be, what more features are needed to be added to it.
Mm-hmm.
So I think use of data in refining science is just so
Mm-hmm.
fundamental that I just regard it as a obvious revolution, right?
Right, right.
That it has become possible today, and it's going to be relentless, and it doesn't take
Right.
anything away from fundamental science. It's just
Yeah,
yet another tool, right, for doing science, which makes us our lives richer, in some sense.
definitely.
And I would
I mean,
just
for
like to
example,
take
we can, if you are inside Dr. Chandrasekhar's company,
Mm-hmm.
you will be getting results all the time, right? So you have tons and tons of data, and you may
Right.
start learning that, oh, it failed in these 5 cases, right? And
Uh,
then you can give it to some data scientist who doesn't even understand the problem, and he can try to identify, what was,
what
why
was
this failed, right?
right,
Yes. So
So
I
right.
I'd
think
just
that's
like
the
to
kind.
take what Professor Arvind was saying as the first example and give you an example of what we have done today. So our first
Mm-hmm.
vehicle is leaving for a very small country called Timor-Leste. And
Uh-huh.
this has a digital X-ray. And the X-ray read is using an AI solution. And
Oh.
anybody who is positive with the AI solution will undergo a test on DroneAct.
Mm-hmm.
So now TrueNet takes maybe half an hour, 45 minutes to give a result, but I can get a digital X-ray result in minutes. So I can screen
Oh,
hundreds of patients and I can pick up only those patients who are likely to be TB positive, and I can
Exactly.
confirm that they are TB positive at a very rapid rate. So I can use this for population screening. Now we are even talking about putting this entire thing in an electronic tuk-tuk and
oh,
going to some of the most inaccessible places that you can think of, and you can do
wow.
population level screening. This is a direct example of what he talked about in terms of TB X-rays, just AI based reading of TB X-rays,
So you,
which
you
is
can
now just
catch
diagnosis.
you can catch TB probably for a person who may not even have severe symptoms, right? I mean, a chest X-ray will show TB
Yes,
or how?
absolutely. Yes, it will.
Wow.
And see,
Right.
you need a very well-trained radiologist to read these chest X-rays. But what AI is
Right.
doing is now bringing the capability of, let us say, the top radiologists in the country or the world directly
Mm-hmm.
onto your platform by
Right.
using appropriately trained data. Of course, with all AI, it's garbage in, garbage out. So your data sets have
Correct.
to be very well trained. That
Right,
model
right.
has to be very well validated. You have to ensure that it is not overtrained. All of those things exist. But as a concept, I think it is revolutionary that you can go and pick up something like this. Not only can you pick up
Definitely.
TB, you can pick up other chest abnormalities because the eye will pick up all of those
Oh,
abnormalities and
yes,
it
yes.
reflects onto other things. For example, somebody may be having fibrosis of the lung. So,
Oh.
for what reason is that fibrosis happening can be picked up. So, tremendous possibilities exist. This is just one example with AI, but so many other
Mm-hmm.
examples in terms of the very large data sets. And as Professor Arvind very correctly said earlier, also we had a lot of data, but there was no way of capturing this data
Capturing, right.
in a structured fashion. Now we have all of the structured data available. So how do you make
Right.
use of the data and do the reverse of building a theorem? You are
Theory,
essentially
right.
predicting based on your pattern. This is what the
Let
human
me,
does. Yeah.
yeah.
Right. So you mentioned training data, right? So training data is very critical for developing these models. So
Yes.
how do we deal with training data? I mean, are the data available? Are they open to anybody who wants to develop, or does somebody have to have access or pay or that? And how do
That's a
you? Are there
very
any challenges?
very fundamental question in an America where people are obsessed with privacy, right? You have to anonymize data, but huge data banks are being built, right? Because individual research do not have enough wherewithal to go and collect.
Right.
Even have access to existing data. So many big institutions, including government, is helping now, right? To collect this. I mean, the data may exist to make it available to people generally,
Right.
right? So, for example, one of my colleagues developed breast cancer, and she's lying in bed, and every time the doctor comes, he says, there is 5% probability of that and 10% probability of that if I do this. So she says, where are you getting these numbers? So he says, oh, this is what everybody tells me, and you can read the papers. So she says, okay, I'd like to read the papers.
Right.
So she started reading all these papers of how these things were being computed. And then she was shocked at how small a sample these
Oh,
people were looking at, right? She says, why are you not looking at every patient in Massachusetts? Why are you not looking at every patient in America? Because as far as we are concerned, even all that much data is not that much data. We can easily analyze it.
right.
And that's what everybody's trying to do now, right? How to collect data in a way so that researchers can have access to it. So I think we are taking it to the next step. It's not just good enough to collect data, but collect data and publish it so
Right.
that people can, analyze it and suggest better and better diagnostics. And just this is applicable in so many different fields, but medicine is probably one of the most useful uh,
Right.
things for that.
Do you see a challenge in sharing this data across geographies, or can people do that right now, or are there any restrictions? So my question is like from people from India, can they access data from other countries, or even how is data managed in India, which is what I'm
Actually,
curious about.
it's the other way around. I mean, for medicine, India is such a rich place, for diseases that, that if India started collecting data properly and making available, as far as I mean, I'm not saying it should be done, but there might even be room for selling it.
Oh my God.
So, no, so I'm
Yeah.
saying that for us, accessing Western data in these things is probably less important than just
Then to
having
analyze
a
our own data.
system of data collection.
Dr. Nair, would you like to comment on
No,
the
I think you have
data?
a point there that India is cagey about sharing any data when it
Right.
comes to these kind of medical records. And I think it is only a question of evolution. I think we will evolve to a stage where there will be some structure around sharing this information. India is, I think, for the first time acknowledging software as a medical device, which
Right.
essentially is an AI-driven system. So these things are starting to happen, and I think there will be methodologies where we can share this data uniformly. Today, it's not difficult to create an AI model of something because everything is available. All the tools are available. You don't require to be a data scientist for it. The question is, what is the quality of data and how do we create a good data bank? And this is something that has to happen.
So do you see a problem in the collection of data also? I mean, sharing is the next step probably,
No,
especially for India.
problem. There is absolutely no problem. In fact, there are so many efforts to try and find out whether somebody has COVID based on an induced cough. So you just cough into your phone, and your phone will tell you that you have COVID or not. Possibly much more sensitive than any other test possible, and it is
Yeah.
not difficult to do. Yes, collecting the right kind of data, having it validated in the right fashion, all of these things are early challenges, and I'm sure we will figure out as a community a method to hasten these kind of things. Any of these things can be data-driven diagnostics.
So, Aarti, if I may just say so, whatever the problem of data collection is, cleaning up the data is equally big problem. So lots and lots of AI is being applied just to clean up the data. And
Oh.
this reminds me of earlier winner of Infosys Prize, Dr. Sarawagi at IIT Bombay, Professor Sarawagi. She was just cleaning up the postal addresses. So if you see the addresses people submit, she says, in India, postal addresses are very strange. They're almost like an algorithm, how to get there. You know? So, so I think I'm sure there will be lots of things about data collection also. Different machines record things differently, different instruments. So I mean, I think there is a lot of work needed to clean up data so that it can be used properly for a machine
For a
learning.
training, right? My next question is, what challenges do you see in defining and implementing the regulations for the use of AI and ML in various fields?
Whew.
Yeah, if
But
it
the
has
regulatory
a,
system is always
sorry.
playing catch up, isn't it? It's the science that takes the lead, and the science establishes the utility, and then regulation follows. And I think that is exactly what is happening in India too. Uh, we are today developing, and we may not get it right the first time around in the sense that we may create something that is a little more stringent or something less stringent than what it should be, but eventually we will get it right. And I think the best part is that regulation has started recognizing all of these advances in technologies and started putting some frameworks in place. So, and that's a very difficult job for them. I mean, when you don't even have a category called software as a medical device, to understand that category, even the US FDA is grappling with it. They've currently moved the extreme left of the pendulum where everything has become so stringent that seeing a software as a medical device it may not come to market itself, and
Yes,
it
yes,
may be
yes.
not having the utility that is required. So somewhere I think regulation will catch up and come to middle ground in terms of what is the best way to protect the patients on which this technology is being applied, vis-a-vis how to get the technology out fast enough for the patients to benefit.
So we see a possible use of AI to fine tune the regulations also.
Yeah, good question.
I think, Aarti, this
Right.
is one of the biggest questions in some sense. I mean,
Right.
as head of CS faculty, I'm trying to expand the faculty in that direction, right? Because there is more and more use of, at least in US, of data for social uh, issues. So, for example, whether somebody should be released from jail, right? So
Okay.
you want to know the recidivism, what are the chances he'll come right back, right, from the jail? And people are studying lots and lots of data, and they can make very good predictions. But these things are all wrought with all kinds of social and moral, ethical issues. The use of AI in these things is cannot be done easily. In medicine, at least, we have a lot of time to test it, right?
Right.
But I think in some other fields, there is not so much time to test the hypothesis whether the automated program is doing well or not. Something as not simple, but not so disastrous as, for example, should we use AI in admissions? And people don't like it, right? I mean, many schools are prohibiting it and so on. So there are lots of ethical issues in this, and I think they are different in different fields. And this will just continue as the technology develops.
So training data has an impact on how whether that solution is acceptable or not. Could be dependent on the training data that is fed to it. Like one example is the Twitter. I mean, last year there was this Twitter thing where the preview always showed the white people and not the black ones.
Yeah, yeah, yeah,
So if
yeah,
a
yeah.
photograph has a white and a black person, then the preview always showed the white person.
Right. I think this is a problem, but it's an easily solvable problem. So, for example, even in face recognition technology, uh, i- currently, and it may have changed already in last 2 years. In the beginning, so for example, it had almost 98, 99% accuracy with white people, and accuracy dropped to 85% for black people, right? And it was much poorer for Chinese, right? Now, if you go to China, their face recognition technology, it does 99% on Chinese people,
Right.
right? So its training data, of course, matters in this and what you are assigning it to and so on. But these kinds of things are understandable and easily solvable. I think the harder part is when there is things going on, you see the result, but you don't understand why it's happening.
Oh,
And then it becomes
okay.
much more controversial
Right, right.
because machine learning is often like a black box, doesn't give you a reason, right? It just tells
Right.
you this is what it thinks. So I think understandability, causality, etc., these are frontier questions in machine learning and AI.
Right, right. Thanks a lot. It was nice talking to you, and it was very insightful getting to know about AI and ML.
Yeah.
And Dr. Nair, it was absolutely exciting listening to your journey.
Thank
And
you. Thanks, Arti.
right,
Thank you. Bye bye.
Thank
yeah.
you.
Thank you for listening. This podcast was brought to you by Research Matters, a SciComm initiative by Gubbi Labs. For more such science-based stories and podcasts, log on to www.researchmatters.in. Until next time, stay tuned.